and are new at the forefront of many vaccine technologies for COVID-19 show that genetic vaccines have not only potential for high potency, low cost manufacturing, and safe administration, but also capacity for rapid development. And these advantages of mRNA vaccines have led to the development of other genetic medicines. And the search for COVID-19 vaccines has accelerated the development of genetic medicine. So I won't go into the detail as we have two esteemed speakers with us, Dr. Lakshman Varanasi, Assistant Professor at the School of Interwoven Arts and Science, KR University of Andhra Pradesh, and Dr. Vaishali Sharma, Visiting Faculty, Department of Natural Science, KR University, Andhra Pradesh, who will share their knowledge and will give deep insight into the recent advancement in biological sciences. I heartily welcome both of you here. I'm grateful that both of you have accepted my invitation and spared your valuable time for my students. I'm glad to find that a large number of participants have joined the webinar, and this large number indeed depicts the popularity of this program, and the credit goes to all the faculty members of biotechnology department, Dr. Soma, Dr. Aruna, Dr. Supriya, Dr. Nayyar, Shalini, and all the other members of biotechnology department. So kudos to all the members of biotechnology department. And a special thanks to Vijeta also, whose contribution in the arrangement of this program cannot be undermined. And I, on behalf of my college, welcome all the participants and especially my dear students, without whom the webinar has got no meaning. And at the end, I wish for the grand success of this webinar and hope that it will prove fruitful and highly beneficial for the students. Thank you. Over to Sophie.
Thank you, madam, for enlightening us on today's topic so elaborately and beautifully. We are highly enriched about this mRNA vaccine which you have talked about. Now I would ask our coordinator, Dr. Shalini Mehta. Has she joined in, Dr. Supriya? I think she had some engagements, prior engagements. She had told one of our faculties that she was pre-decided appointment was there.
Yeah.
Hello. So we would continue.
Is not there.
Okay. Okay, Supriya. So I would like to invite Dr. Supriya Srivastava, Faculty of Department of Biotechnology, to introduce our first speaker, Dr. Lakshman Varanasi.
Thank you, Dr. Somarai. Good morning to all the respected dignitaries present, my colleagues and dear students. It is with great pleasure that I would like to welcome Dr. Lakshman Varanasi. Dr. Varanasi has done his graduation in biochemistry from Osmania University, Hyderabad, post-graduation in life sciences from GNU, and PhD in biochemistry from University of Mississippi Medical Center, USA. He has postdoctoral experience in USA, Austria, and Czech Republic. Dr. Varanasi has worked as Head of Life Sciences Research, Pratibha Healthcon Private Limited, Hyderabad, as Chief Scientific Advisor, Biomedicine Sri Kalika Technologies Private Limited, Hyderabad. At present, he is working as faculty in Department of Biology, Division of Natural Science in Kreya University, Hyderabad. He has multiple publications in eminent journals to his name. He enjoys yoga, writing, and programming in Python language. On behalf of Department of Biotechnology, Ranchi Women's College, I would like to extend a warm welcome to him, Dr. Lakshman Varanasi. Sir, please. I think he's not there.
Dr. Varanasi, Dr. Vaishali. Hello, Dr. Vaishali.
Yeah, I talked to him. He said he will be joining soon. Just give me a second.
Yes, he has joined. He has joined.
He has joined.
Okay.
Most welcome, sir.
Good morning, sir.
Good morning. Most welcome.
Lakshman, you're mute.
Good morning, everyone.
Hi.
Thank you.
Good morning.
Good morning.
Should I keep my video on or should I?
Yes, sir.
Go off?
As per your convenience.
Thank you.
You will be sharing your screen, sir. Dr. Lakshman.
Yes. I'll be sharing. So, okay. I thought there was something else, some other events before the actual talk, or should I start right away?
Oh, right
If
away.
I can.
Yes.
Okay. Okay. So give me just a couple of minutes and I'll be with you.
Our system coordinator, Mr. Ali, has just messaged that we are also streaming live on YouTube. Hello, Dr. Vaishali. Sir has.
Okay, do excuse that delay. So should I go ahead and share my screen?
Yes, sir.
Okay, and can you all hear me like properly? And just
Tim's kindly response.
Yes,
Yes.
it's audible.
It's audible,
Thank
sir.
you.
Yeah.
Thank you. Okay, I'm going to share my screen then. Okay. All right. So thank you again for having me. It feels kind of nice. Feels very nice actually, because I'm actually I was born and raised, and I was raised in Jamshedpur in Bihar, and so thank you again for inviting me. I have fond memories of Bihar and Jharkhand. I just wanted to mention that I'd like this to be a presentation where where the students get something out of. I hope they get the most out of it, and I would like them to ask questions. Should they not understand something, even if it interrupts the talk, because that I feel is the more important aspect of this. Also, you don't have to wait till the very end, because if you don't understand one thing, you may not understand the next thing. Right? If you feel too inhibited, please feel free to type up the question and send it over by message, and I'll try to get to it whenever I take a break between the different modules. Right. So to jump in right away, I'm going to be presenting work that I did a few years ago, but which I feel is still relevant, and it helps me highlight some techniques, especially in proteomics, that are very relevant today. And I want to go through the workflow of the project, the work I did there, and then sort of try to highlight certain techniques and certain complications, certain issues in the practical, in the actual experimentation for this work. So we're working on the biomarkers of neoplastic disease and the early diagnosis of cancer. And you'll see in every slide, we'll have the questions, hopefully, from them. So essentially, this was the question. So how can we detect early on a cancer of the gastrointestinal tract? Because how can we detect the cancer when it is easiest to remove or destroy? What method should we employ for this? And there are obviously other ways of doing this. This is the procedure we took on. So of what types are these cancers of the gut? They comprise the duodenal, the gastric, the hepatic, the pancreatic, and the colorectal. And occasionally we also include the oral cancer in this list. So this is just an illustration of what I just mentioned. We were especially interested in the pancreas. We were interested in the gut as a whole, but we started off with the pancreas because the typical feature of these cancers is that they are very insidious. Right? You don't see them until it's too late. That's one reason we actually took it on ourselves to go after this. So why do we care? The incidence of cancers of the gut and the resulting mortality is a compelling reason. The mortality from these cancers from these diseases is really horrific. This is a schematic of a representation of the incidence of I think this is for colorectal cancers, as you can see here. The darker the blue, the greater the incidence. And as you can see in this part of Europe, Central Europe, where I was working, they have the highest per capita incidence. They have the highest incidence of this type of cancer in the world. And so it was an especially compelling reason. The mortality, the map for mortality resembles this. It's very similar. So early detection can improve survival. There have been studies which have proved that early detection can improve survival. Whether through any medical intervention, or through any other means of handling it. What is a biomarker? It's useful to sort of understand what a biomarker is before we get any further. It's any characteristic that is objectively measured and evaluated as an indicator of a normal or a pathological process, and or a response to a therapeutic intervention. Because then you can measure, especially in the last case, you can measure the efficacy of the drug, right? So we all know of some biomarkers. These are temperature, body temperature, skin color, eye color, body aches, blood pressure, and so on. These are all what we call gross symptoms or gross biomarkers. But it is quite often the case that the disease does not manifest itself through gross symptoms, right? It's hidden, and that is precisely the case with these cancers. And so we need to move from the gross to the molecular marker. It is essential. Right? For instance, we could look at, say, an increase or the decrease in the level of a particular protein, or the number or the type of chemical groups it bears. And all of these are candidate biomarkers, really. So this is a step, take a little perspective to this, get some perspective to this. It takes about 12 years or more for a cellular lesion to become visible to imaging. The lesion must have about 100,000 cancer cells before the lesion becomes visible. And the growth isn't linear. You can have a lesion that sort of grows for a few years and then stops, and then continues again after a few years if it receives certain stimuli. Sometimes the tumor is latent throughout life. Sometimes the tumor grows for some time, then it just completely stops. Right? And we don't know what it is that makes a tumor behave this way. It's a non-linear fashion. And as I mentioned, to reiterate here, the cancer, especially that of the gut, is insidious. Normally, for instance, that of pancreatic cancer, the 5-year survival is the 5-year survival is less in the single digits. So the rate is in the single digits, and it's really abysmal. Because we know that blood flows through all parts of the human body, it is likely to carry signs of the body's condition. It constantly reviews the body's condition. And so if you could get a snapshot in terms of a blood sample, then at any given point of time, you know what the condition of the body is, or we should. But we have yet to learn to read the signs in the blood properly. So the blood could contain dead cells, secreted proteins, hormones, metabolic waste, and others. And so, it follows that proteins from tissues, including tumor tissue, drain into the bloodstream. And so this is an important premise for any work that we do. Mathematical modeling. This was shown a few years ago by this group which did a simulation. They saw that in this long period, when the tumor grows, there is a window where the tumor is not visible to imaging, but it has started leaking proteins into the blood. So if you could sample blood and analyze it properly, then you would be able to tell early on if there was a cancer. You would catch the cancer at a stage when it was manageable and when intervention would help. So I didn't talk about this, but so what is the current standard of care for diagnosis? How do we detect and diagnose cancer today? And really, we don't have many tools to detect cancer early on. We really don't know, especially because the symptoms don't manifest themselves. Right? There are a few, the blood-based markers, CA19-9, the carcinoembryonic antigen, bilirubin. CA19-9 is not always, you have a lot of false positives. You also have, when you use this biomarker, you also have problems because non-neoplastic diseases like, say, pancreatitis would also occasionally throw out this marker. So you have to be careful when interpreting the results from this. So what is the best source for a biomarker? Ideally, something that you can analyze or sample easily. Right? In this case, blood. Right. So let me again digress here, just to talk about post-translation modifications, such that we can understand the process better. So we all know that genes code for RNA, and then RNA codes for proteins. And once synthesized, the proteins may be subject to further modification by enzymes. These are chemical modifications, and these are called post-translation modifications. And there are a myriad ways in which these modifications can happen. So to show you a schematic, there is a polypeptide coming from an mRNA. There's a folded protein, and then a protein with chemical modifications. And I've shown them in different shapes because the different shapes represent the different types of modifications. And these proteins carry information in these modifications. What is the type of modification? What is the number of modifications? What is the arrangement of these modifications on the protein? All of these carry information. They have a syntax there that we need to understand. The one that we're especially interested in is glycosylation, because these are typically the modifications that indicate a progression, a neoplastic progression from lesion to tumor. Just to do a sound check before I go any further, can you all still hear me properly?
Yes, sir.
Hello?
Yes, sir.
Sorry?
Yes, sir. Yes, sir.
Yes,
You're audible.
sir.
Okay. So knowing all this, one of the hypotheses we can put together is certain glycosylated proteins in the peripheral blood, meaning in the blood that you can draw, are indicators of developing neoplastic disease or cancer. Right? And so the question becomes like, which proteins are differentially glycosylated as a manifestation of neoplastic growth? And that question is really an important question. It's worth a lot of money as well, literally. So how do we go about identifying these proteins that are glycosylated? How do we isolate them, identify them, study them, and then determine which ones we should look for in a patient's blood? There are problems here. I mean, the very first problem is that blood is a very complex mixture. There are more than 10,000 proteins that comprise the blood protein profile. And the bigger problem is that they don't just all exist in the same concentrations in blood. There are some at really trace quantities, and the most abundant protein is about 10 to the power 9 times in abundance as opposed to the least abundant. Right? They span 9 orders of magnitude. This creates its own problems, but one that's manageable. So what is the solution to such complexity? You would want to simplify the sample before you analyze it such that the interpretation, the analysis is more precise, more accurate, and you can interpret something meaningful from the analysis. So there have been preparatory procedures that have been developed for different types of protein populations, meaning populations of proteins with different modifications. So I want to switch to a different document just to show you, right? So here's so this is an analysis showing, we show the blood without without such fractionation and blood with fractionation, or more precisely, serum, murine serum, on which we are doing the experiments, really. So you can see the first is just a marker. The second row and the fifth row, I guess the sixth row actually, they represent serum from mice. And if you run serum from mice and from humans, you'll see that it's very similar. And you see this large band here and here, they comprise mouse serum albumin. So the most abundant protein in blood. More than 70% of blood is serum albumin. The 5 most abundant proteins in the blood will comprise up more than 90% of the blood proteome. Right? And so this becomes a problem because they mask the proteins in small quantities. And these are really trace quantities, right? So once we do the enrichment, there's a mechanism. There is a method called it's a device from Biorad called the Proteome miner. It removes the 5 or 10 most abundant proteins. It'll remove the proteins in a manner that is proportional, right? So if they're most abundant, they'll remove so much. If they're least abundant, then they won't remove anything, right? And so they balance, they sort of level out the dynamic range of the protein concentrations. And you can see in this column and in this one, column 7 and column 3, that you see a lot more proteins, right? And this is because you've removed most abundant ones. Now you can see more deeply into the proteome. And just to, I should have mentioned at the beginning, this is essentially a gel, an SDS page, polyacrylamide gel electrophoretic run that has been stained with silver. So silver staining is very sensitive to proteins present in small quantities, as opposed to other staining techniques. If you see, and if you can read, I hope you can see these numbers. So, for instance, when we do this experiment for the human, we see how many more peptides or proteins we see when we apply this enrichment technique. 160 as opposed to 36, or 267 as opposed to 162, right? And if you look at this, because we had these peptides together in the mouse, and I'll come to that in a few minutes, but more than 1,000 as opposed to 400. So at least approximately three times the enrichment. And this is fabulous, right? Because you want to be able to look deeply into this. And so maybe I should. Right. So let me just talk about this. Let me go back to this, and then I'll switch again. So every time you want to do a study, you need to use a model. Right? So and the model we chose was the xenograft model, the murine xenograft model. Murine meaning belonging to the mouse, and xenograft meaning an external adventitious extraneous tissue that you source from any species you want, in this case human, and insert it into the murine tissue, murine body. And so this requires surgery. So a mouse is grafted with a human tumor or with a mass of human cancer cells that are grown in the lab. And there are two reasons why we do this. One is that any human proteins you see in the murine blood must essentially have come from the human tumor. Also, the murine blood volume is about 1,000 times less than the human blood volume. And so it is that much more likely that you will see these particular proteins, the proteins coming from the tumor. And so this is the workflow. We engraft the tumor in the mice, we let it grow for about a month. It reaches a size of about 1 cubic centimeter. We harvest it, we prepare it, we analyze it. And this shows you the workflow again. And once you do this process here, we get the serum, which we put through a process called the solid phase extraction of glycoproteins, which is an enrichment technique that I spoke of. And then you put it through a mass spectrometric workflow. And then among the list of proteins you get, you need to be able to discriminate between the human and the mice peptides, because you were really interested in the human peptides. So I'm going to switch to the other, just to give you an illustration of the process. So this is the original paper that described this process. It's a Nature Methods paper, the solid phase extraction of N-linked glycopeptides. These are really peptides, proteins that have been, peptides are really, the way it's done, the way they're created in the lab is by cleaving, by digesting proteins with the digestive enzyme. Right? We normally use trypsin, you could use chymotrypsin, and it sort of eats up the protein in the sense it breaks the protein into small pieces. And these are the pieces which then bear these different these glycosyl groups. Right? So here is an illustration of that process. So this is a solid phase matrix, normally silica beads, or sapphirose beads as well. So it shows you some of this chemistry, and it's quite elegant, the chemistry. So we know that if you've done some biochemistry, you will know that monosaccharides have a reducing group at one end. They have an aldehyde group, and so they can undergo a condensation reaction with hydrazine, right, and form a hydrazone, or in this case, it can form a hydrazide bond, which in this case immobilizes or binds the protein, or peptide, to the protein, to the resin. So this is the bond that I'm talking about. Maybe I should go further. Right. So this is the hydrazide bond. This is the glycosidic bond. This is the bond that connects the glycosyl group with the protein, right? Or in this case, the peptide. Now, the glycosyl group, it doesn't have to be a monosaccharide. It can be an oligosaccharide. It can be a much larger oligosaccharide. It can be a polysaccharide. And so, but really what we're interested in is the reducing end at the monosaccharide, that is the terminal group on this glycoside. That's what binds to the resin, right? And so this is also a motif, the NXSD motif on the peptide, where these glycosyl groups are essentially bound, right? And so, and this becomes essential when you put the data that you get from mass spectrometric analysis to discriminate between mouse and humans, or mouse and human peptides, and to be able to tell if a particular peptide was really glycosylated. So we digest this with an enzyme. So once we process all these proteins from the samples, we will once we've had them bound to these solid supports, we basically run into a column in a high pressure liquid chromatograph, and then we get these different peptides. what happens when you cleave this particular bond with this enzyme, it's called PNGSF, is that you get a peptide that is one Dalton like lighter, and the aspargine to which the oligosaccharide is normally bound is been converted to an aspartate. It's a very important part of the chemistry, especially in the bioinformatics of it. So let me just go back to the right. And so this is how the data looks, right? Once you've done all the analyses, you've put all these, we've processed the blood from the mice, we've done the mass spec analysis. This is what the data looks like. You can see that we did a lot of these cell lines. So we grew up all the cell lines that represented all the different common cancers of the gut, and they're all listed here. And the axis at the bottom represents the group, the cancer group. For instance, the first one is the colon cancer. So all of these represent colon cancer group. Then the esophageal cancers, the gastric cancers, the liver cancers, pancreatic cancer, and so on. We also had a control group here, and then we had cancers. We had samples where we had actually put in tumors from human patients into mice. Right? You can see that the liver cancers yield the most peptides, right? And what does each dot represent? It represents a protein that you detected from that particular sample, right? And the more intense, and this is a color scale showing how confident the process is. If the dot is more towards the red, then it's present very abundantly. If it's dark blue, then not so much. Right? But we don't try to get rid of any information at this stage. Once we have this information, so now from this we knew what all peptides we could expect to see in a person suffering from any of these cancers, right? So the next part of the process was to actually look in patients with pancreatic cancer to see if you could actually see these peptides. This would be the validation. Right? You wanted to see, and so not only could you see like which of these peptides are present in a cancer patient's blood, you can also measure the levels. That's an important part. Just to go back, how am I doing on time? And if you want me to like wind up in a few minutes, or
No, sir, it's okay.
yeah, it's okay.
There will be a question answer session also. The students might like to
Absolutely. Is there anything? Does anyone have any questions right away, or should I just like go through the whole thing?
Students, please feel free to ask questions.
Yeah, please feel free, guys. Yeah.
You can put it up in your chat box also.
Right. Mm-hmm. I guess because I can't see the chat box when I'm sharing the screen, I'll check it occasionally because I would really like you to understand. Is the process so far clear, sort of?
Student, please ask your doubts.
The results. Okay. So, okay. So this is essentially, okay, maybe I should show you the, okay, so that's not there. Okay, so let me explain this process. Maybe it'll become a little clearer. Okay. Okay, so right. So this is the workflow, really, right? So we grew up cancer cells in the lab because our institute is an institute of translational medicine. We were also able to procure or obtain tumors from the hospital. In fact, that's one of the reasons the institute was set up. To be able to help the doctors see better, be able to diagnose the disease better, and to be able to study whatever if there's anything new that the doctors saw, they would tell us about it. Then we would try to analyze it, work on it, and tell the doctor what we thought. So we had these cell lines. We grew up about 3 different cell lines, each cell line representing cancer cells for a particular type of cancer. Either it could be a colorectal cancer, or pancreatic cancer, or a gastric cancer, a hepatic or a liver cancer. So we grew them up. Once they had grown, we would sort of scrape the cells out of the tissue culture dishes, and then we would mix them with Matrigel, which is sort of a jelly substance, and then insert that particular lump subcutaneously in mice. And these are mice that are typically immunodeficient, so they don't reject the tumor. Right? The tumor starts to grow in the mice, and we wait for the tumor to grow for about a month or until the tumor acquires a size of about 1 cubic centimeter. Right? Once that happens, we sacrifice the mouse, we harvest the mouse, we exsanguinate the mouse, meaning we collect the blood, we cut out the tumor, and those two things constitute the base samples for this part, for the actual laboratory workflow. And in this case, we just worked on the serum. We didn't work on the actual tumors, but we had some data as well from it. I'm just not showing it here. So once we had the serum, then we could go to work. Right? We put the serum through the process of the solid phase extraction, where we enriched the samples for glycopeptides, right, for glycoproteins. Once we had enriched them and they were ready to go into the mass spectrometry workflow, right? So, the mass spectrometer doesn't exist by itself. It's always coupled to a protein, analytic device called the liquid chromatograph. What the chromatograph does is it separates this huge lump, this huge mix of peptides, glycopeptides, and it resolves them, separates them according to their hydrophobicities. Right? So those that are least hydrophobic flow out first, and then so on. Because we elute them, we elute them from the it's called a reverse phase column. Essentially, we try to resolve all these different peptides such that we can look at those peptides one at a time in the mass spectrometer. Maybe I could pull up a picture.
There's another question, sir. Two questions are there.
Sure. Okay, so this is a. I'll take a look at the questions in a second. So this is how it looks like, right? Or maybe. Right. So this is how it looks like. So this is the mass spectrometer, and this is the liquid chromatograph. So you load your sample here in the chromatograph, and then it separates all these hundreds of thousands of peptides according to their hydrophobicities, and then they're introduced into the mass spectrometer like one at a time, right? So then they come in from here into the mass spectrometer. And this entire process is automated. So all you need to do is purify your peptide or your sample and put it into the liquid chromatograph, and then you get a readout. There's a bioinformatic analysis as well. I won't go into that. There's a bioinformatic analysis once the mass spectrometer has finished with analyzing all the peptides, and then you know what the list of peptides are that you got from the mass spectrometer. Okay, maybe I should go back to the. Yeah, you said there were two more questions. Or the mice transgene. Yeah, okay. How do we diagnose cancer? Anu has asked a very pertinent question, and patient for evaluation. Okay, good questions all. Yeah. Anu, that's a $1 million question, really. Let me just finish with this, and then I'll come to Preeti's second question. So, Preeti, these mice, they're actually, they have been genetically altered, which is why they cannot launch an immune response to a perturbation, to a challenge. So they are genetically modified mice. We call them, I think it's called severe combined immunodeficient mice, SCID mice. They've sort of improved by several iterations now. We call them humanized mice now, and you can tailor them for whatever you want to work on. But I'll come to that. Yes, so they are genetically modified mice. Go back to your first question. I just want to make sure I've covered what you were asking. Right, and so once you analyze this list and you distinguish the mouse mutant peptides from the human, that's when you would get this. That's how the data looks at the end. Okay. Of course, you did the analysis as well. Okay, to go on to the Anu, I'll come to your question closer to the end, where I actually talk about it. Well, I could mention some things here. We do have the standard of care we use in the hospital right now is imaging, different kinds of imaging. One kind can actually, sometimes we'll feed the person a tracer molecule and look for radioactive emission from the tumor. Of course, the problem is with all these techniques, the tumor has to be of a certain size for you to be able to see it. Right? And this is, with the gut cancers, it's really, you can't tell early on. And so this, right? How can we detect the cancer even before they're visible to imaging? Right? Some cancers will have some cancers will actually have and will manifest themselves in gross symptoms like earlier than others, and in those cases you can obviously tell. Pragya is asking why we cannot use the tumor cells directly from the patient for evaluation. Well, that's one way of doing it. So normally, so this actually answers Anu's question as well. So normally when a person goes in for a check and we feel he or she has a cancer, we will do a tissue biopsy, right? We put a small biopsy needle into the tumor if it is accessible to the surface, or you send we call it a endoscope, and endoscopes can also sample tissue, And then once you have that tissue sample, the biopsy needle, that is we send it to a pathologist who will then stain it and check to look for certain histopathological markers for the presence of cancer. Right? That's the normal way. But it's usually, you have to have enough cells for the pathologist to see.
Please mute your mic. Please mute your mic.
Okay, and so we do use the tumor cells directly from the patient for evaluation. Sometimes, for instance, the pancreatic cancer is the pancreas are located deep inside the person's viscera, the abdomen. Sometimes it's difficult, depending on the location. Sometimes if the tumor is closer to the bile duct, the endoscope will pick up can pick up the cancer cells. Otherwise, you can't. Right? And so that becomes an issue. What is the need to enrich cancer cells with glycopeptides? Okay, so I realize I wasn't very clear here. We're not enriching cancer cells with glycopeptides, right? We have grown the cancer cells, the human cancer cells, in the mice, right? You put the human cancer cells into the mice. You have waited until it becomes a tumor. You have removed the tumor and the blood from those mice, and then you are trying to enrich. So, okay, I guess the word enrich may have misled you. So they're trying to improve or increase the concentration of glycopeptides in the blood, not by adding more glycopeptides to the sample, but by removing other constituents of the blood. So if you remove phosphopeptides, unmodified peptides, sumoylated peptides, sialylated peptides, then you're left with only the glycosylated portion of the proteins, right? Okay, so that's how we're enriching or sort of increasing the proportion of these glycopeptides in the sample. I shouldn't have used the word enriching, I guess. What are the parameters that determine a competent immune system? A competent immune system gives a response to cancers after immune induction. Okay, I'm probably keep that question for later. I mean, you have to have if you have ablated or removed, for instance, we call it the lymphomyeloid lineage. The lymphomyeloid lineage in the bone marrow is what gives rise to all these immune cells, all the cells of the blood and of the immune system. Now, if you mess with those cells in the bone marrow, then it is unlikely that you will get a proper functional immune system. Right? I'm not sure exactly how the SID mice, what exactly goes into creating these immunodeficient mice, but obviously they are messing with those very parameters that you're asking about. Right? So there is no immune response. Right? There should be a part answer to that, Sonali. What are the ways to treat mitochondria? Okay, so I think we're digressing. I'll still try to answer your second question, Sonali, but let me keep it for the end of the talk. Okay, I'll come back to your question. So, should I go on, Soma, or?
Yes, sir. Yes, sir.
Okay.
You can.
So I'll just
we'll have a question answer session after
okay.
your talk.
So we'll take off. Okay. So we'll do that. Okay. So to go back to the maybe I should talk about this process a little more. So these are the results. Remember, this was the first set of results was from the screening step, right? You just wanted to know overall what all the peptides or proteins were that indicated the presence of cancer, right? Now we want to go further and check for these particular proteins in patient blood. And so the process we take up, we use is called single reaction monitoring, or SRM. But we need to do some homework before we get there, right? Before we can actually do this validation. So the first question, because we're biomedical scientists, we want to know who else has reported these first, right? So we took the list of proteins that we had, we ran it past the literature, and then we found that there were some that corresponded with the ones in the literature. So the ones marked in brown indicate the ones that we saw in the literature, right? Again, maybe I should just skip this. Okay, so to come to this technique, right? This is a technique for measuring the abundance of a protein analyte in any given sample. It can be a plant sample, it can be an animal sample, whatever. Right? So in this case, for this particular method, a peptide is a substitute for a protein, right? And it is what we call the reagent or the SRM assay. So, and this goes into the very nature of the peptide. Right? So any given peptide, so a peptide is essentially a small sequence of amino acids. And each amino acid has a specific mass, which means each peptide has a specific mass. Each peptide also has a specific charge. And this ratio of mass to charge is unique for every given peptide, right? For a given peptide or amino acid, the mass to charge ratio is constant. And this is the basis for the analysis that we do by this method, right? So once we have this peptide, we pass it into the mass spectrometer, and the mass spectrometer fragments it using high energy electrons. So you get a peptide fragment. Now, this conversion of a parent peptide into a fragment peptide by the application of a certain amount of energy is what we call a transition. And a transition, like a peptide, is very unique to that particular peptide. It's very unique. Now, sometimes you might not get just one fragment, you might get multiple fragments, right? So 2 or 3 or 4 or any number. And each of these, and the conversion of the parent peptide to this fragment is very characteristic of that particular peptide. We call this conversion of parent peptide to any given fragment, we call them a transition, right? And so the idea is if you knew of at least one transition for these peptides that you identified in the peptides that we saw in that list, then you have a chance of measuring its quantity, because this is very distinctive. This is very unique and very peculiar to each peptide. And if you have a peptide that's very peculiar to that particular protein, and then you know that doesn't occur in other proteins, then you know that you have a solid reagent. You have something that you can use for measurement. And so when you look for the signal for this particular these fragments in the mass spectrometer, you'll see something like this. You'll see 3 peaks, or you'll see as many peaks as there are fragments. Right? You'll also see a peak for the original parent peptide, because you'll obviously have some of it that doesn't fragment. So again, this is just to show you how the mass spectrometer works. So initially, you're blowing in all these peptides from the liquid chromatograph, all the peptides that are being dissolved on the basis of their hydrophobicity. And then you apply, because they have charge, you can apply a voltage gradient and select just one particular parent peptide and let that go into the next chamber in the mass spectrometer. We call it the Q2 chamber, right? That's where you fragment the peptides, right? And then you can choose a particular fragment from among the different fragments. You can select each fragment like one at a time by varying or by varying the voltage. So you cycle the voltage through a certain set of values, and every time it goes to a different voltage, a particular fragment comes through, right? And then you see this particular graph. So now, if you have these graphs, if you can see a proper peak for a particular peptide and a peptide fragment, then the area under the peak gives you the abundance of that particular peptide or fragment, right? And so once you know what the intensity of the peak is, you can extrapolate to the abundance of the protein, the original biomarker protein in the blood, right? And for its presence, obviously, they're present. So, but you can't use any peptide, right? The peptides have to go through a certain scrutiny. They have to pass certain qualifying, so they have to have certain qualifications to be able to be considered as a peptide useful for quantitation. And so these are the candidates. And so in our case, we identified about 476 from the original screen, from that plot I showed you, where you had the different dots from the different cancer cell lines. We have to make sure that these don't have cleavage sites. Did the enzyme used for the original digestion of the protein cleave it completely or not? If it hasn't cleaved it completely, then we throw it out. It shouldn't have methionines because methionines get oxidated. They get oxidized. And oxidation causes a change in the mass of the peptide, which means it is no longer unique, right? You're getting some artifact. The peptide length must be within a certain range. You can't have peptides that are less than 5 amino acids, or peptides more than 25 amino acids. You want them in a certain range because then they work well in the mass spectrometer. There you can fragment them nicely. They must be unique, right? If you see the same peptide from two different proteins, then you know that you can't use it because the peptide is then not a surrogate for that particular protein. Quantifying that peptide won't tell you anything, right? You don't know which protein it's from, right? And so once a peptide passes all these different criteria, we will call it a monotypic peptide, meaning a peptide that is a proper reagent for the SRM analysis. Right? Now you can use it to measure that particular protein in the sample. And so sometimes you don't have to, the peptide that you're interested in already has these coordinates. So the transition, the mass to charge ratio of the parent peptide, the mass to charge ratio of the fragment peptide, and the energy required to fragment the parent peptide. These are what we call coordinates. And so these coordinates are already available in an online repository called the SRM Atlas. There are some that are present in There are some that are present in another repository called the Peptide Atlas. And so you don't have to create these coordinates by yourself. So you can use these coordinates available on the web, program your mass spectrometer with these, and then go ahead and put your sample in, and it will tell you exactly how much protein you have. Right? So it turned out that of the 400 or almost 500 peptides we had, 12 did not have coordinates. So we went about measuring them in our own mass spectrometer, and I identified all these different peptides, SRM assays. Right? So that was I should maybe right. And so from the 500 that we had, how many did we have to work on ourselves? For how many did we get the coordinates from the SRM Atlas? It's just a list showing that. Yeah. So let me just go back to the original control. Okay. Right. So where do we go from here, right? So now, so I've just gone over this part. This is what we call the validation phase. We identified a set of glycosylated proteins in the original sample, and then went forward with it to try to develop the assays required for measuring them in the patient serum. So the purpose of this study was to come up with a classifier. The purpose of this study is to come up with a panel or a device that can distinguish between people who have cancer and people who don't. Right? So so that's the purpose of all this work. And we didn't get to that part, but our own group was working on a different project at this time, and they were working with colorectal cancer, and they came up with a group of about 5 proteins, a panel of 5 proteins that could distinguish between people with cancer and without cancer. It could also distinguish between people who had stage 1 cancer or stage 2 from people who had stage 3 or stage 4 cancer. Right? Yeah. It was at this stage that we were sort of thrown off course by a problem. And this is typical of when you're working in the experimental sciences. Things don't always work. The instrument may not work. And so we ran into something that was really an intractable problem. And we just couldn't figure it. And then, but we're going to talk about that some other day. So I'll be happy to, that's all I have for now. I'll be happy to take any questions you might have. If you want me to explain some things, I'll be happy to pull up certain pictures. Maybe before you get started, let me just show you. So this is the instrument really that we used for the validation. So this is the instrument that we would use. This one actually. So this is it's called a Q-Trap 5500. This is the instrument that will tell you. I mean, it has an accuracy. I mean, the manufacturers claim that it has a limit of detection or limit of quantitation of about the order of nanomoles, picomoles. We've never had to go that far. But it's a very useful property because typically the proteins that are secreted by these cancer cells into the bloodstream are present in very trace quantities of the order of, say, 10 to the power -9. Just wondering if I should, yeah. So I'll be happy to take any questions. How can we diagnose the stage of cancer? So, Anushri, so this is something that we hope the panel can do, right? So hopefully the panel is sensitive enough. So for instance, you can just consider one protein, for instance, right? If you just saw one protein that you knew was present, that the cancer, the tumor secreted, right? Now, imagine that this particular protein is, the concentration of this protein is very sensitive in the blood, right? It's very sensitive to the size of the tumor. Right? So now, for instance, you have, say, 5 persons, and each is in a different stage of cancer, hypothetically, right? And so in each stage, or in each person, you would see different quantities of this particular protein. Right? And so that's how you would be able to tell if, which stage of cancer each patient was in. Now, typically, we don't normally use just one protein because the chances of error are very high. So we'll use a panel. So we have proteins A, B, C, D, and E. Protein A goes up, B goes up, C goes down, D goes up, E goes down. And so if the degree to which these have gone up or down in a particular person indicate, the stage of the tumor in that particular person, then you have a nice panel. That is really what we're aspiring. That is what we're really shooting for, really. That is the ideal panel that Okay. Uh, so Anushri, yeah. So yeah. Okay, I'm gonna. Sonali, just remind me to like go back to your question at the end. So Kriti, name of the proteins used in detection of states. Right. Kriti, if I knew, I would be worth a lot of money. I don't know, right? This is what we're trying to figure out. Um, of course, well, I'm not authorized to release the list of proteins that we detected in the experiment because that is copyrighted. That's intellectual property. But there is no dearth of candidates, really. I mean, the literature actually has hundreds and hundreds of candidates that have never been properly validated in patient serum, patient blood. We have all these candidates, but the standard of care still uses a particular biomarker that we discovered decades ago. The actual, and this highlights one big problem between discovery and actual clinical use. If you cannot show that the proteins, the biomarkers you identified are better than the current standard of care at detecting or identifying the disease or the stage of the disease, then the clinic does not have any use for this. They're not. You have to show that what you create as a biomarker for a diagnostic device is better than that which is present in the clinic. If it is not, then they don't care for it. And so, where the biomedical community has really failed is transitioning these candidates to the clinic. Right? For a long time, we didn't even have the technology. For the past 2 decades or decade and a half, we have had the technology, and it's only now. And I should probably show you that particular slide now. It's the right time to show you that. So, for instance, this is a paper by Rudy Ebersold, who's really been the pioneer in quantitative mass spectrometry. How do you until a decade and a half ago, we just had qualitative mass spectrometry. The first half of what I spoke about, right? You just knew that these proteins were there. We had really no way of a method where you could identify the quantity of the protein or peptide, right? At high throughput. This one does both. This is SRM is still like a decade old, right? Now you have what we call data independent acquisition, or DIA, where the mass spectrometer will really tell you all the proteins that are there. So it's a detection as well, and the quantities of all these proteins, right? Not just one. We're talking about thousands here. It works, right? Now it's become a problem of analysis, right? I think we're limited by patient cohorts, so we need a large enough patient cohort, to be able to validate these as well. The other problem is more of a social problem. You want to be able to screen patients. It doesn't help if you try to do this after the patient has an ache in his abdomen, right? Because he or she may already have cancer. You want to be able to do a screening of people at risk, say at the time of they're 45, and then get the data for all of these proteins, and then tell if any of them is at any risk for developing cancer. Unless you do a screening analysis, no one is really going to show up for this kind of analysis until they actually feel some symptom, right? Some ghost symptom. But this was a funny paper where they said, for a long time, the Western blot was really the gold standard for quantitation. And then Rudy suggested here that we could actually, they were getting requests from these journal editors who were asking authors to validate their SRM data using a Western blot. And Dr. Abersol argued that this is meaningless because the SRM is so much more, accurate than the Western blot. And so it's time to stop asking, asking for validation of mass spec data using the Western blot, right? But so to go back to the question, right? I think so. Priti, I think I'm just gonna stop there with your question. How do tumor cells transmit their epigenetic? Maybe I should epigenetic features to daughter cells and maintain the malignant phenotype. Right. So if you look at epigenetic features typically, well, that's actually a good question. I'm not sure, so I confess I'm not sure how the epigenetic signature in a parent cell is replicated in a daughter cell. So yeah, I'm sorry, Manali, I don't have the answer to that question. Normally, when you have a growing tumor, you don't really need to have, because the tumor is the result of about 6 to 9 mutations. Right? These are fundamental mutations that need to be present. You can have many more mutations in the tumor cell above and beyond these 6 to 9 mutations, right? And so, even without, I would think, even without an epigenetic, copying or photocopying, you would still have a malignant phenotype. Now, when the tumor is growing, you'll have these different populations in the tumor. We call them clonal sectors, and each has a different set of mutations, right? And each of these sets of mutations renders that particular population malignant. But I really don't have answers to how they transmit their epigenetic features to daughter cells. Okay. What conclusions are made if a novel protein shows high intensity in experiment? So a higher signal intensity will imply a higher abundance of the peptide in the sample, which in turn will indicate a higher abundance of that original protein from which the peptide was derived in the patient sample, or in this case, the mouse sample, the murine sample. How can we know that the mutations that cause a patient's cancer shape treatment? We don't know right away, right? We're getting into the realm of precision medicine with this question, really, because patient's cancer shape. I can give you an example, for instance, right? Maybe the example will illustrate this. So initially, going into it, we don't know. We know some things from experience. So if you take, say, triple negative breast cancer, which is the worst form of breast cancer. Okay, let me go to the one where you don't have all 3 mutations, right? So the triple negative breast cancer is so called because there are 3 different mutations happening in 3 different cell surface receptors. So imagine the least harmful form of breast cancer, for instance, right, where you don't have any mutations in those 3 proteins. Right? So normally, the breast cancer tumor in the breast grows because of the estrogen receptor, the ER receptor. The estrogen hormone binds to this receptor on this tumor cell, and the tumor cell multiplies and so on. one of the treatments that we use for this kind of tumor is it's an antibody. Yeah, it's an antibody therapy where it goes and binds to this receptor and blocks the binding of this hormone to the receptor. So the hormone to this receptor, the tumor is made to slow down. The growth slows down, and you sort of have the patient can live longer. Yeah, or at least it gives you the time for other interventions. What happens in triple negative breast cancer is when the epidermal growth factor receptor, the EGFR receptor, in the ER, obviously the estrogen receptor, and there's one more, I think, I forget, there's a third receptor as well. All of these three are mutated, meaning you cannot use an antibody therapy to block these receptors and cause and stop the tumor from growing. So the tumor is resistant to these particular drugs, right? And so now we know from experience that these won't. Well, the purpose of precision medicine is to identify other such mutations that are rendering the tumor resistant to the therapy, right? And these days you never give like just one therapy or all therapies at once. They'll give you some therapy, then they'll stop it and give you some other therapy, just to make sure that the tumor doesn't become resistant to all these therapies at once. But now the idea of precision medicine is to identify all those mutations that can help you understand whether the person is resistant or will be receptive to the drug. So when a person goes into the hospital these days, they'll do a screen of the mutations right away because they don't want to waste any time, giving you a therapy that won't work. So that's one answer to that question. Shows shape treatment. What is the technique to block transmission of epigenetic features to daughter cells? I'm not sure. I mean, I like the interest in epigenetics here, but I don't have an answer to that. I mean, I wish I could invite some colleagues to talk about epigenetics. I guess maybe this is I'm so sorry. So, Ria, I'm sorry, I don't have an answer to that question. I mean, epigenetic features are features that happen above the genetic level, right? So these are acetylation which normally will block off, will shut down a particular region of the genome. So I mean, you don't even have any cellular expression there. Yeah, I don't have an answer to that. Yeah, I'm sorry. Okay, so Suhani has a question.
Excuse me, sir.
Yeah, tell me.
Because this gene expression in epigenetic modifications, like DNA methylation and all, but they are not affecting the genome sequences. So I think that inheritance of epigenetic will not cause any danger to that person who is inheriting, because it doesn't actually change the sequence. It's the environmental changes. Can you highlight? Another question of mine was, have you tried these Gleevec, all these medicines like anti-cancer therapy like Abraxane, Doxil, Gleevec on your proteins, the recent which are being given for chemotherapy, all these proteins which you are studying? Have you tried all these?
So we didn't use any drugs on the mice. If that's what you're asking.
Yes. Okay. Thank you.
That would actually be a good experiment. I mean, we want to see how the profile of those proteins changes, how the protein profile changes.
Yes, that's why the current chemotherapeutic medicines, if you allow those medicines to be treated to those protein, it can extending of your work.
Yeah, that's a good question. Yeah. Yeah, we didn't try that. Should maybe try that. I mean, yeah, there's an issue. I mean, it's still a good experiment. I mean, what you're suggesting, when the mice have a tumor that's about 1 cubic centimeter, these are really like small mice, and they're already quite sick. One of the things I would worry. I mean, so I would probably try to do your experiments in maybe rats or in mice with smaller tumors, because once the tumor grows, yeah. But that's something that would be.
Thank you.
Suhani has a question. Let's suppose a female is suffering from first stage of ovum cancer, but the male sperm is not on that condition. Is fertilization possible? So you're talking about ovarian cancer here. I'm not sure of. So I'm not sure of if the ovum is not healthy. I would think so. Fertilization can probably still happen, but you would have mutations in the zygote if the ovum is also cancerous. I'm not even sure of whether the ova from a woman suffering from ovarian cancer are cancerous. I'm not even sure if it is wise to have a pregnancy when person has ovarian cancer. Yeah. So fertilization, I would think, is still possible, but I think the zygote would have mutations that, or the embryo would have mutations. Yeah. That's a good. I'll have to think about that more. What is the current success rate of cancer treatment? Really, yeah. Okay. Let me go back to Sonali's question. Further back up. Sorry about that, Sonali. He had two questions. Yeah. What are the ways to treat my type and stage of cancer? Yes, is there any risk to treatment was given? No. So what do you mean by my type and stage of cancer here? Sonali? So is Sonali there?
Sonali, you can unmute your mic and ask, sir.
Sir,
What are the ways?
he is asking many type of cancer.
Oh, any type of cancer?
Yeah, many type of.
Okay, many types of cancer.
Yeah.
So there is always risk here. Yeah. So really, it depends on what type of cancer is it. I mean, if a person had, say, if a man had, say, we call it cancer of the prostate, right? Versus colorectal cancer. The cancer of the person has a greater chance of living much longer with the prostate cancer than with the colorectal cancer. There was a study where they autopsied about between 50 to 100 men after, obviously, dead after death, obviously. And they found that a large number of them had the prostate cancer, but they never had the symptoms that would sort of impel them to go have a checkup done. They actually lived their whole lives without the prostate cancer becoming a problem. Colorectal cancer, these are very insidious, very pernicious cancers. And with all the drugs that we have available, you can still die. And just to give you some illustration of how bad these are, I know I've heard of many doctors in the United States or elsewhere where once they realize they have cancer of this sort, they will refuse all treatment because not only does it, so the treatment really spoils the quality of life that's left available to you. And so, so they just refuse the therapy. They just ask for painkillers, right, until they actually pass away. Some drugs, and now more and more of these precision medicines have become, excuse me just a second. More and more of these precision therapies have become much better. Because very recently, I think a couple of years ago, that we got these, the immunotherapy came into prominence. So essentially, what you do there is you let loose, you try to let the immune system, you trigger the immune system to go fight the cancer. So you have far fewer side effects with this kind of precision therapies than you do with conventional chemotherapy, right? And the chances of survival longer are far greater with the precision therapies, right? The problem with conventional chemotherapy and radiotherapy is that it kills all cells, healthy and cancerous. Yeah, I mean, of course, even with immunotherapy, for instance, they've seen. This goes back to another question that was here. So it's not. Let me go back to. So it doesn't work for everyone. If you have a certain set of single nucleotide polymorphisms or differences in your, if you have a certain mutation or polymorphism in your genome, then these therapies may not work. Immunotherapy may not work. So even in a group of say 100 people, the immunotherapy may work in about 30 of them, but it won't work in others. What is it that makes these 30 responsive to the therapy and the others and the others resistant? We don't know. There's so much that we don't know, right? And so this is where we're slowly trying to use more and more intelligent ways of analyzing the genome to see like what gene or non-coding region as well does what. Right? We don't know. Right? Hopefully, in the near future, we'll come up with some more answers. But Sonali, is that? Let me go back to what to say. Right. So to go back to this question, what are the ways to treat many types of disease? Typically, you can treat a cancer. So if you we were at this session with venture capitalists back when I was in Europe, and so we were trying to present, among other works, my research as well, and the idea was to convert this panel of markers of peptides into. So the idea was to advance these to the clinic, and so the venture capitalists came over, and so they asked me like what my work does, and I told them that these peptides, we're trying to see if these peptides can identify a tumor in the early stages. And right away he asked, like, which stage? And I said, hopefully the second stage. And he said, if you can't identify, if your peptides cannot identify the cancer in the first stage or earlier, then it's of no use in the clinic. Right? It's no use to us. Right? So if you can detect a cancer in stage 1, stage 0, stage 1, or maybe even sometimes stage 2, then you have some chances of excising or removing that cancer and saving the patient, or putting the patient on some drugs, killing the tumor. The problem when you have advanced stages is that the tumor has started to spread. So cells from the tumor have started to spread to other parts of the body, and they've established secondary foci of these cancers. And these are typically the ones that kill person, right? So you have hepatocellular carcinoma or pancreatic carcinoma that is spread to the brain, to the lungs, to the bone marrow. And really, you can't fight. There are too many. You can't fight all battles. Right? That said, there have been like many instances of cures that were never expected, right? So there's still hope. Somehow some people pull through. And again, it goes back to genetic factors that we really don't have any clue about. Sonali, I think I'm going to, yeah. It also depends on the person's, constitution. Was the person like very healthy before the cancer? How well is the person able to fight or survive the treatment and the cancer, right? So I think those are the features. I think if you have a follow-up to that, Sonali, I'll be happy to take that question. Okay. Okay. Will immunotherapy be a turning point in the fight against cancer? It will be a turning point against certain cancers. Uh, yeah. I'm very sure it will be, so you're right, it will be a turning point. And as we'll probably find more and more use for this therapy for more and more types of cancers as time goes on. Yes, it will be a game changer. I mean, the nicest thing I find about these precision therapies is the fact that you have such few side effects. What is the most common cancer? Amisha's question. Amisha, it really depends on which region of the world you're looking at. The Czech Republic and other countries in Central Europe have the highest incidence of the cancers of the gut, colorectal, gastric, hepatic. In India, among men, it's oral cancer because of tobacco, and across men and women, it's lung cancer. There's a very high proportion of Indian women suffering from ovarian cancer as well. That's a big problem in India. So it really depends. It's a contextual question, that Misha. Can blocking the production of abundant glycosylated protein help in disease symptoms? No, really, the glycosylated protein is an effect. Well, actually, I think that's a good question. Actually, I take that back. We don't know if it's an effect of the mutation or of the lesion, or if it's something that causes further problems. Yeah. In many cases, so in many cases, it is the result of something gone wrong. If you have a glycosylated protein, very often it's an indication of something aberrant in the cell. So, but blocking the production of the glycosylated protein is unlikely to help in disease symptoms because it's already there. And by blocking the production, if you're talking about drugs that prevent the expression of it, I don't think it will help because the mutation is already there. Kavita Kumari, can we overcome tumor resistance to platinum-containing antineoplastic drugs by activating the sensitivity factors in the tumor? Yes. Yeah, platinum-containing antineoplastic drugs comprise part of the old school conventional therapy to cancer, and they have a lot of side effects. I don't have the answer to this question. One of the approaches to these therapies was to provide, to make the cells more susceptible or sensitive, like you mentioned. But yeah, that's beyond me, Kavita. I don't have the answer to that question. are the latest methodologies useful in the cancer treatment? Pretty successful. Pretty successful in saving life patients. Yes, I mean, the ones that we have certain bright spots. For instance, let me give you another example. That's a bright spot, and this goes to cancers of the blood, thalassemia, leukemia. One of the reasons you have these diseases is because of mutations or problems in the progenitor cells in the bone marrow, the the hematopoietic stem cells in the bone marrow. Now, there's a way now of reconstituting. So you reconstitute using the person's own stem cells from his or her own umbilical cord or from someone else's umbilical cord. So you get the stem cells from these places, you put them into the bone marrow of the affected patient, and you wait for the patient to develop normal blood cells, right? And so that way, the percentage, so you ablate or destroy the cancer-containing cells using radiation, and then you put in these good cells, and so the number of healthy cells outnumber vastly the aberrant cells, and that helps in the therapy. So you have some success there, especially for blood cancers. Immunotherapy will slowly spread to other cancer types as well, is what I see. But immunotherapy is one subset of these precision therapies that are happening, probably the most successful so far, but there will be others. So can cancer be cured? Again, this is very contextual. Can cancer be cured in all cases? No, but hopefully in the future. Which cancer is the rarest? Just to give you an example, we had an artist friend at our institute in Europe, South African, and she actually was an Ashkenazi Jew. Is one of the sects of the Jewish population. To give you some history, the Ashkenazi Jews were the ones most persecuted by Hitler during his assault offensive against the Jews in Europe. And so my colleagues' grandparents on both sides, they escaped. They were from Estonia originally, and they escaped to South Africa. And one of the problems with the Jews in Europe, the health problems with the Jews in Europe, was that of inbreeding, right? And so many of these, congenital problems were passed on to their progeny because of so much inbreeding. You see the same thing in the Amish population in Pennsylvania, in the United States. And so this girl had mutations in her genome, and she had this benign growth in her kidney. And the growth was benign for about 7 years or so. And then it became malignant. So they did a genetic analysis of her genome, of her tumor cells, and they found that she was just one among 7 reported persons in all over the world who had that particular mutation. Right? It didn't even have a name. It was just some cancer of the kidney. So which cancer is the rarest? I would say this one could be one of the rarest, not the rarest. But I don't know to give you an exact answer. After the chemotherapy, is there any chance to repeat cancer again in the human body? Yes, this happens quite often. We call it a relapse. And you see this happen quite often, especially the tumor has spread. So you can always find, so there will be some population among the scores of populations in the tumor that render a particular population like resistant to that therapy, right? That's the problem. So it may kill like 50 of the 60 populations, but then you'll have 10 populations in the body that are resistant to that particular therapy. And so you'll still have, and so they'll grow again into a tumor. Sometimes they don't, sometimes it's very successful. Just mining the blood proteome. Right, so Usha's question, what can we conclude from this after mining the proteomes from urine blood? What information do we get? We get a list of proteins that are candidate biomarkers for the early detection of cancer. Of course, the question then is, this was a question that was put to us by our collaborator. He's like, guys, you're looking at full-grown tumors in the blood, right? How do you know that these same proteins? So we got a list of these particular proteins, right? How do you know that these proteins are also there in people who have a smaller tumor, right? So we don't know. We decided we needed to start somewhere. And so we tried this, but ideally you would want to look at the tumor in different stages of growth and then collect the proteins and do the same analysis. Then you'd see. So, what is the life expectancy of patients suffering from stage 4 metastatic gallbladder cancer? Really, I don't have an answer to this, Twinkle. Yeah. When it's stage 4, it's basically, the most full-grown metastatic gallbladder. If it is metastatic, I wouldn't have, I don't want to be the, the spread doom and gloom here, but yeah, I wouldn't say very much time. I wouldn't say it's very long. What bothers me is the metastatic part, right? If a tumor could grow and not metastasize, I wouldn't worry about it too much because you can just remove it. But once it goes into other organs, it goes into the bone marrow, goes into the brain. I don't know. I don't have the exact answer to that, Quinto. What are the benefits and risks of each of these treatments? Well, that's a very broad question. The benefit is that it kills part of the tumor, the cancerous growth. The risk is that you also kill a lot of healthy tissue. And I remember, from personal experience, the symptoms of these people on these therapies, these conventional therapies, and the side effects are just horrible. It's just horrible. They're nauseous, they're sick. I'm talking about chemotherapy and radiotherapy. Yeah. So that's a very broad question. I don't have an answer to all of those. If parents are suffering from cancer, then what are the chances of getting cancer? Really, so the good news is that mutations are not heritable, right? Unless it's a germline mutation, right? So generally, if you have a cancer, then it's unlikely that your kids will also have the cancer, right? Here's the other thing. So, for instance, there are certain genes in a person, and these are not inherited, right? There are certain genes in individuals that are more prone to mutations than other genes, right? And the classic example, the most recent ones was that of Angelina Jolie, who had a mutation in the BRCA gene, and the risk of her developing cancer was about 70% or maybe 80%. And once she had the surgery done, the risk of her developing cancer went down about less than 10%, I think. But normally, if a parent has cancer, it doesn't follow that the kid will have cancer. That's not the case. Yeah. Unless there's a mutation in the ovum or the sperm. Actually, and this goes back to another question. Actually, so mutations are stochastic. So there was a paper in 2015, I think, where they said that. Excuse me, let me just stop sharing. A flag mutation. Any mutation is the result of a stochastic or random event. Right? It's a random event, and you don't know who will develop cancer and who will not. You can lead a healthy life all your life and then develop cancer. Sometimes you can smoke all your life and not get cancer all your life. Sometimes, and so really, so the idea of what they suggested in that paper was that, what they proposed in that paper was that it's a random event, but you can increase the likelihood of that event by smoking or drinking too much, right? Or even a little. Yeah. So if parents are suffering from cancer, then what are the chances of getting cancer, right? I think is there any pills for the treatment of cancer? Yeah, Sonali, if you can qualify that question, I'll be happier. Yes, there are pills for the treatment of cancer. What treatment do you recommend? How do you pronounce your name? This question here. Is it Miss Alameen? How do you say your first name? Well, anyway, I was just curious as to how so that I could address you properly.
Mahzabeen.
I wonder. Excuse me, say that again.
Mahzabeen. Mahzabeen.
Is it? You'll have to be specific here. Yeah, what treatment do you recommend? Like for what cancer? And I really confess, I'm not. I probably still won't be able to give you a good answer because I think much better to ask a physician. We sort of know of treatments when it comes because we use them in our research as well, but I wouldn't have the answer to that question. Yeah. But did you have any specific cancer in mind there?
Cancer.
You didn't. Okay. You'll have to unmute yourself and tell me. Did you have any specific cancer in mind there?
It's liver cancer.
I'm sorry, I can't hear you, but that's. Yeah, I wouldn't know. I'm sorry. What is the main chemical compound present in the pill? Sonali, you'll really have to be more specific. I really don't know. Sometimes the medicines will be antibodies. Sometimes they'll be chemical compounds. Sometimes, yeah. There are a variety of drugs. That question is very specific to what drug you're talking about. So I don't have an answer to that. If a pregnant woman has cancer, then can that woman pass cancer to the baby? Again, if there is a germline mutation, meaning a mutation in the ova, then I would think she can pass on that cancer to the baby. But otherwise, other cancers are not heritable. You won't. I think you cannot pass on a somatic cell mutation to a child. It doesn't happen. Hardest to detect. Oh boy, I think I have to take a break and get a glass of water or something. I'm sort of losing my voice here. Yeah, excuse me if you can give me a 5-minute break, I'll be happy to come back and answer some more questions. Soma,
Yes, sir.
is that okay? I'm going to take a 5-minute break. Yeah, I'll be happy to come back and answer your
We will
questions.
be collecting, the students will be collecting their questions. They will write it down and be ready for the
Absolutely.
questions.
So, thank you.
Yes, sir. Thank you, sir. Students, you can write it down and keep your questions ready. Sir will be joining us in 5 minutes again. We have sent you sir's email ID in your group. Later on also, if you have any questions or doubt, you can remain in contact with sir through his email ID. Hello, Dr. Vaishali, are you there?
Yeah, hi Suma. Yeah, I'm here.
Good afternoon. So, your session, you can kindly tell us about the exact timing, because we are on time, exactly on time.
Mm-hmm.
When will you start your session?
Just as it's mentioned in your poster.
2 p.m. Okay.
Yes.
Okay. So our students Riya, Rani, and Anjali, please send the link at 2 p.m.
Is this not the same link?
I don't think so. Riya is there. Hello. They are managing the link.
Okay, no problem. Send
Yeah.
me 10 minutes before.
Yes. Riya, can you talk to ma'am
Yes, ma'am.
about that link sharing?
Ma'am, the new link is posted at 2 p.m.
Ma'am is asking you 2 minutes before.
Hi Ria, could you just send me like 10 minutes earlier, like maybe 1:45?
Yes, ma'am, no problem.
Thank you.
Thank you, ma'am.